Good evening, this is Mr. Wintermelon. So we have learned a great deal about the transport of water in xylem. In the last video of this chapter, we are going to learn about the transport inside the phloem. And the process is known as translocation, and it occurs in the phloem. It is the transport of dissolved nutrients. So by the end of this lesson, you should be able to define the term translocation as the transport of food or dissolved food in phloem and illustrate the process through translocation study, which basically means that they can come up with some example and you should be able to say, this is evidence for translocation in the phloem. So translocation is the transport of manufactured food substances such as sugar and amino acid through the phloem. Phloem transport is bidirectional, meaning it can go up as well as go down. So from leaf, it can be transported to the flower that is on top or the fruit that is on top, as well as get transported to the root. But just bear in mind, when we say transport sugar, or in this case sucrose, as well as amino acid, we are not transporting them in their solid form. They are dissolved before they can be transported. So there are three case studies that you ought to know. First is the use of aphids. I have mentioned this in the first video of vascular bundle. Second is the use of radioactive carbon isotope. So what is radioactive carbon isotope? This is more of a chemistry term, but for your understanding, just take it as carbon molecules that we can use device to detect their presence because they are emitting out some kind of signal for us to detect them. And lastly is the bug ringing experiment. So for the use of aphids, what the scientist does is, aphids will be attracting phloem sap by inserting their proboscis into the phloem. So you can see the aphids, they will poke their proboscis into the stem. Then scientists will do something very mean to these aphids. They will numb the aphids with carbon dioxide. Then they will chop off the body with laser. And what will happen is the phloem sap will flow out from the proboscis due to the hydrostatic pressure. And when we analyze the phloem sap, we found sugar. In this case, is sucrose. So from this, we can tell that phloem is used to transport food, since analyzing the proboscis, which is poked into the phloem, detects food. As for radioactive carbon isotope, what they did is they will pump in carbon dioxide that are radioactive. Radioactive means they are sending out some signal. Then it will be given to the plant, so plant will undergo photosynthesis and use the radioactive carbon dioxide to make into glucose. So they will check section of the plant and look for location whereby this radioactive isotope can be detected, and they were found in the phloem. As you can see from the diagram, is mostly on the outer area, while there's no radioactive carbon isotope that can be found in the xylem. So phloem is used to transport the glucose that was produced. And lastly is the ring bug experiment. So just try to recall where is the position of the phloem relative to the xylem in the stem. So phloem, right, is on the outside while the xylem is on the inside. So if we just chop off the stem, the outer layer of the stem that contains the phloem, what will happen is when the plant is transporting the sucrose, to, let's say, the root, it actually cannot pass through because the phloem channel have been severed or have been chopped off. So what will happen is, instead of depositing the sucrose into the root as such, the plant will just deposit at the lowest point, that means just before it was cut, and you can see the stem that is swelling as a result because starch had been stored at the area. So this will be all for the chapter of transport in plant.